But that's okay, because now we're done with the homework, and we're going to go ahead and do some of the new stuff. Okay, please remind me. I always forget. Evolution. Hey. Evolution. What does evolution mean to you?
Let's check it out.
Yeah, what does evolution mean to you? Pop it in the chat. Right on our screen is totally fine. You can write in the box, or if there's not enough space, you can write anywhere else. What we're going to kind of really emphasize is that language in science is really precise, right? So we use words deliberately, and we're going to continue to use words really deliberately, and it's really hard to be deliberate, and so sometimes we make mistakes, and then we have to go back and like refine what it is that we mean, right? You're popping in all sorts of really great words: variation, change, adaptation. One of you drew, I think, a turtle. It's awesome. Adapt, growth, Darwinism, survival of the fittest, trial and error, really good. Natural selection, excellent.
Yeah, and there's a bunch of words like advance and favorable.
Yeah, yeah, favorable traits.
Interesting to see. We're going to talk about those.
Yeah, we're going to talk about those super specifically. Simple and constant. You know what, though? There's an interesting trend, though, that really focuses on the word change, and I think we need to pull that word out, especially when we think about the word evolution, because a lot of these terms that you've shared are part of the big, underneath the big umbrella that is evolution. So things like natural selection, things like adaptation, things like advancement, things like favorable traits, all of those are terms that fall under the umbrella of evolution. But when we talk about the word evolution, precisely what we mean is what most of you have written down here, is change. Not necessarily in a good direction or a bad direction. There's no value added to it yet, but rather just the idea of change. What do you want to add to that?
Yeah, the idea that advance, that's a common one that probably you've been taught, and it's there's change for sure, but advance, that's one of the things we're going to get to, we're going to pull out of you, and we're hopefully going to advocate for you pulling it out of friends and family when you hear them talk about it.
That's right. Yeah, exactly. So what we mean by change, like what's changing, what we mean by change is often at the level of the DNA, the allelic frequencies, or the frequencies of the alleles that are represented within the population. You kind of use that as the marker for figuring out whether evolution is happening. So it isn't about whether something, adapted or whether something mutated. It's more about whether it resulted in a change in the frequencies of alleles. Now, this evolutionary biologist may very well have a different scale of defining it, and that's totally fine. We have to be precise when we talk about it. Mm-hmm. Are you an allelic frequency kind of person? No?
It can be.
Are you like a mutation person?
Mutations are everything.
Mutations are everything. So Smith here would look at mutations, and if he saw mutations, would be like, yep, evolution is happening, right?
Evolution could happen.
Right.
Yeah.
And somebody else might look at it and go, well, okay, there's a change in the allelic frequencies in the population, evolution is happening. Whether or not it's natural selection, genetic drift, or gene flow, those are sort of mechanisms that happen sort of underneath evolution if we're going to make a big like concept map. Possible things that can happen that describe the direction of change, and we're going to be focusing on those for the most part. So not necessarily improvement, not at the level of evolution. The only thing that we need to see for evolution is change. Whether it's possible that it will improve the situation, right? And that would be natural selection, but it's possible that it wouldn't necessarily improve, that it could be for random reasons, and that would be genetic drift. And we're going to like bust open all of that over the next couple of days. But first,
why would you study it? And realizing that in some parts of the world you have to defend why you would study it. Mm-hmm. I can't figure out where I have to sit here in this arrangement.
Sorry. You're all uncomfortable.
Yeah.
Okay.
So why would you study it? Well, you're Lots of reasons, and we're living through one of the big ones. The application of evolution to medicine, to the study of and the prevention of disease, to diseases jumping, and this last with David Quammen's book, Spillover, to diseases jumping between species. These all are underpinned or supported by an understanding of evolution. And in fact, that's a whole new branch of medicine that you could get into if medicine and human health, even veterinary medicine, epidemiology. Epidemiology is an inherently evolutionary kind of a discipline. So one of the cool ways we like this is a visualization from a science paper. It's several years old now, but it's so beautiful to me in terms of demonstrating for you change within a population in response to some kind of selective criteria. So something in the environment. So what's going to happen here is you're looking in that YouTube screen at a big agar plate, and we're going to inoculate it at left and right sides with bacteria. And the agar plate, we've built it in strips like parts of a hockey rink, and behind the goal at either end, there's not a lot of antibiotic. And then as you get closer to center ice, there's stronger and stronger. There's more and more concentrated antibiotic. And so the bacterial hockey players are going to have a tighter, a harder and harder environment in which to live. And so let's watch what happens.
So what we ended up building was basically a petri dish, except that it's 2 feet by 4 feet. And the way we set it up is that there are 9 bands, and at the base of each of these bands, we put a normal petri dish thick agar with different amounts of antibiotic. On the outside, there's no antibiotic. Just in from that, there's barely more than the E. coli can survive. Inside of that, there's 10 times as much, 100 times, and then finally the middle band has 1,000 times as much antibiotic. And then across the top of it, pour some thin agar that bacteria can move around in. The background is black because there's ink in it, and the bacteria appear as white. First, you see they spread in the area where there's no antibiotic, up until the point they can no longer survive. Then a mutant appears on the right. It's resistant to the antibiotic, it spreads, until it starts to compete with other mutants around it. When these mutants hit the next boundary, they too have to pause and develop new mutations to make it into 10 times as much antibiotic. And then you see the different mutants repeat this at 100. And after about 11 days, they finally make it into 1,000 times as much antibiotic as the wild type can survive.
As you're watching this, perhaps you're seeing evolution in action. What you're seeing is mutations in action. Those mutations we can then physically map on this agar plate. I think one of the other things that you're doing is probably, if you've ever been a patient in a hospital, you're going, oh, that's perhaps why I wasn't prescribed, or the antibiotic that I thought I should get right away, or why the doctor made a really emphasized to make sure I take this until the end of the course. And it's because of the strength of mutation. So when you asked about alleles, this to me, that video is, it's mutations. Now, why also might we use, what other ways might we use evolution? There's a whole raft of ways. This is maybe a non-intuitive way. This is using phylogenies. And so you're looking at phylogenies of plants from 3 different regions: in the South African Cape on the left, in Nepal in the center, and New Zealand on the right-hand side. And the tips of these trees have been color-coded according to which ones human societies in each one of those 3 regions have used for medicinal purposes. Why is that interesting? Well, we can map. We're using the tree as a map, and in this case, a predictive map, because what we can see are cases, yeah, exactly, like this, where there's a whole cluster of that tree, a branch of that tree, where a lot of the taxa there, a lot of the species are used, but there's cases where there's some that aren't. From a pharmaceutical perspective, from a medicinal perspective, that's hugely suggestive that it's like, let's find and run some trials for different things on those taxa that we haven't used yet. So we're using the phylogeny, the evolutionary history plotted in a tree diagram to predict where we might find things to be useful to us in the future. And we're all living through this. This is a video we made way back in
the day.
Yeah, back in March of April of 2020.
Isn't it March 700th today?
It is March 700th, 2020. So congratulations, the
Yay.
longest month. But even then, this marching through this kind of great visualization of the diversity of the viral genome for COVID-19 and how it spread and how, so this is all alpha actually. All, like, if we were to remake this map now, this is not current enough that it includes Delta or Omicron.
That's right.
So you might be one of the most tree familiar generations to take 1070 ever, simply because of visualizations like this that have kind of run alongside of our understanding of COVID.
Yeah, and I mean, it was evolutionary phylogenies that demonstrated to all of us that Omicron has been in the world since August 2021, at least, right? Solidifying the fact that it isn't that South Africa created those conditions, it was that they have super rockin' scientists that discovered them. And really kind of set us on a course for understanding how it is that the virus mutations and variants evolve and how they spread. There we go. And of course, from an ecology perspective, or from an environmental perspective, or a natural resources perspective.
Agricultural, forestry.
Yeah. All of these things really matter. So, you know, if our fishing practices target larger fish, you might think that you would be changing the allelic frequencies in the populations that don't get targeted, right? And what we've actually been able to see quite readily and for quite some time now, that fish populations are getting smaller. Like, their bodies are not growing as big because we're fishing out all of the big ones that have these, you know, mutations and alleles or these traits associated with being larger bodied. So all of these types of activities, human activities, have a real and demonstrated effect on the evolution of species. And speaking of evolution of species,
Are you mussels?
Are you mussels? Okay, so we really hope that you didn't mind going through some of the stuff for the first, and don't forget to do the second week by Monday, reading and the quiz, because we're going to be focusing on freshwater mussels of the Great Lakes. So based on the things that you've read already, we are going to sort of highlight some of those words that we've already kind of introduced to you. So the first one is endemic here. Okay, an endemic is a very precise term. Mm-hmm. It means that something evolved there and that it is not anywhere else. And so there and anywhere else needs to be defined when you talk about it, right? So it can be endemic to the Great Lakes. It could be endemic to Lake Ontario. Those things are different, right? They mean different things. And more importantly, if they're endemic, it means that they're not found elsewhere. Okay, so one of the ways that you can determine whether something is endemic is to look for it elsewhere and not find it. So we talked a little bit about endemism, and we talked about the endemic groups of mussels or different species of endemic mussels, the unionids that live in the Great Lakes. And we have a question for you on Menti. Oh, hold on. There we go. I'll just launch it for you, but you can already start reading it and thinking about it. So what factors contribute to the threats? If you want to open up a web browser and go to www.menti.com, I will launch that question right now. What factors contribute to the threats to endemic freshwater mussels? Some of you are already putting it in. That's great. Go ahead and just throw those in. Excellent. I don't know. Okay. We've got 240 of you. Just give it another few seconds. Excellent. Well done. Okay, so let's take a look at this word cloud. It's really good. I think you can see that, right? Excellent. Okay, so clearly invasive species is popular. So is zebra mussels, because that's, I think, the invasive species that we're talking about. But pollution and climate change are there. Agriculture is there. Super good. Fishing, erosion. Some people are more specific in their use of terminology with reference to zebra mussel. So zebra mussel may actually be much larger than it is. Runoff, excellent. Okay, worms, frog kept on popping up for a while. Amazing. Okay, so yes, all of these things are important things to take into consideration when you're trying to understand the biodiversity of the Great Lakes with respect to their mussel populations. It's fantastic. Now let's think a little bit about which of the following variables may not be relevant. So we brainstormed a whole bunch of different relevant ones. Let's think about which ones may not be relevant. So I'm going to stop sharing that one and I'm going to throw out the next one at you right now. Take a look and see which of the following are unlikely to be relevant to explain, and again, we're being precise, right? The biodiversity of mussels in the Great Lakes. I think Daniel's awake. Do you need that again? Excellent. Just another few seconds. Most of you have already shared your answer. Okay, 10 more seconds. 5, 4, 3, 2, 1. And remember, if for some reason your tablet doesn't allow you to actually click, it's not a problem, but commit to an answer, because I'm going to end it right now and I will give you the answer right away. So actually most of you did get the correct answer. Daily fluctuations in weather are not relevant to describing or to explaining the biodiversity of mussels in the Great Lakes. They are relevant, but just not to this particular topic, right? They might help to explain the daily cycling or the daily movements or the eating habits of mussels. They may be all sorts of, related to mussel natural history, but they wouldn't help explain the biodiversity of it, of the different species that's within the lakes. Okay, great. So we're going to skip this one because we have committed to blowing your mind with freshwater mussels, and we kind of want to spend some good time on this. That was just a little exercise to, again, get you thinking about different things related to the mussels, but this is cooler.
Yeah, to get you thinking about things about the mussels, but also to model for you the kinds of questions that you'd see in any kind of an assessment in the class. Cool.
So,
mussels.
Mussels.
So unionid mussels, the greatest diversity in the world for unionid mussels. That's a particular family of mollusk, of bivalve mollusk. Is found here in eastern North America. It used to be historically, and now we are chipping away at it. The unionid mussel, many species in that family have a life history that models a bunch of this. So there are larvae that are launched into the world by the adult mussel that stays rooted in the substrate. That's at the bottom at 6 o'clock on that diagram. You can see a blow-up of one of those larvae. So that's not appreciably the same in scale, but a blow-up. So they have a very specific kind of a Pac-Man style larva called the Gluckitia. The Gluckitia is going to feed on, it needs a host, a fish species, and some of the specificity of that relationship between mussel and fish is beautiful and jeopardized by the human transformation, the anthropogenic transformation of Eastern North America. They then drop out of the fish, the juvenile settles, and so it basically tracks this lifestyle. But we're going to talk about some of that specificity that's there between 9 o'clock and 12 o'clock.
That's right. We're going to focus on the unionids, okay? And then we're going to challenge you over your homework to focus on the zebra mussels and their group, right?
Because zebra mussels are not unionids. They are a mussel, but they are not this group that lives here.
So the really important key point and the difference for the unionids is this aspect of the life cycle, that they produce these Gluckitia larvae, not veliger larvae, and that they have to pass through an association with a vertebrate, like just a totally different group of organisms in order to survive. This is their natural cycle, right? And then it becomes pretty challenging for something that doesn't move to plant their larvae into something that is pretty active in terms of movement. And the way that unionids do this is the mind-blowing part. So imagine you're a mussel, you're sitting there on the bottom of the lake, and you're like seeing these fish swimming by, and you're like, oh, I got to get my larvae into those fish. How do I do it?
And remember, by seeing the fish,
they're
sensing the fish because they don't have eyes.
Right. So they do it in multiple ways, right? And that what contributes to the diversity within the unionid mussels. There are many different strategies, not just one good way of doing it. And there are costs and benefits associated with each strategy.
So they're essentially fishing, right? And if you've been fishing, if you're a fisher yourself, you know that in your tackle box, you probably don't go out with one set of flies or one set of lures. And unionid mussels have also, through evolutionary time, evolved a tackle box. So let's take a look at some of them.
Okay.
So they can physically trap. The adults can snap themselves closed on, that's in the bottom left-hand side of the adult fish. Why would they want to do that? They are not eating the fish. They do not want to eat the fish, but they are trying to pause the fish so that that nice bloody fresh gill material is very close to them, and then they can spit the Gluckitia larva onto them, so the Gluckitia snap onto the lamellae, to the gills of the fish.
The next type is called the Conglomates, and these blow my mind because these are the fishing lures that Dr. Smith was talking about. So they make fishing tackle on these long lines, and then at the end have these little packages of the Gluckitia.
It's a piñata.
Inside of things that look like the food of fish.
Yeah.
Okay, those are the Conglomates.
Yeah. So they fish with those Conglomates, and you can see that in C in the bottom center. Those Conglomates are then lured out there as the fish kind of nibbles it, poof, puff, and the piñata of little Gluckitia then adhere to their gills. They also transform their mantle. The mantle is an elaboration of the body, of the interior body, so inside the shell. It's actually what makes the shell. It's an incredible feature of mollusks. It can do practically anything given enough evolutionary time. So in this case, it's building the shell of the mussel, and it's also elaborating into a lure that looks like a fish. That looks like not the species of fish they're trying to put their Gluckitia on, but the species of fish that species of fish wants to eat. And it's doing it when it has no eyes.
Monstrous. And then finally,
what?
Broadcasting Conglomates. These guys are just like, screw it, I'm just putting them out into the world and they're going to stick to a fish somehow.
Yeah.
These are the different strategies.
So I hope your mind is blown. If it's not blown, we can do better than describing it.
Let's show you. Let's take a look.
Take a look quick at the Gluckitia. This is from the text that we use in 2700. That's the hook, the little trigger in the center. Boom, and all of that white material, those are all the Gluckitia larvae. So tens of thousands of larvae, and that's the fish going.
right? You don't want to kill your host.
That's right.
Don't
And you've
do
been
that.
I've seen some of you in the chat talking about this. This does not, it's successful when it does not impede the respiration of the fish. This is not going to kill the fish.
No. Check it out. But those are the Gluckitia that are stuck. And
it's not a tumor.
And the gill creates these little cysts around it, right? It's kind of a like immunoresponse that kind of just provides all sorts of warmth and protection for the Gluckitia and nutrition to be able to develop and grow. Okay. Ready?
More.
More. Show us more. Okay.
Look at those things.
These are the conglutinates. So now take a look. This is like one of the little sacs that these things build, that the mussels build, that get packed with all sorts of Gluckitia. But the little packages, these are lures that look like fish. So check it out.
Just imagine if this was at a child's birthday party and there were little parasitic things.
And he's like, that's not food. But you can see some of the Gluckitia.
Yep.
Yeah, there you go, coming out of the gills, right? He's like, oh, gross. Yeah, I've been infected.
Oh, fish.
Oh, fish. There we go. Yeah.
But you know what?
What?
There's more.
Oh, it gets weirder.
This is the grip. So you can see their elaborations, those photos on the left-hand side. That's the mussel. That's the edge of the shell. And so this is not the mussel eating the fish. This is the mussel essentially French kissing the fish and putting the Gluckitia into, just into the gill tissue.
Okay. Yeah, the fish is like, somebody help me.
Somebody help me.
Another one's going, hey, dude, I just ate a conglutinate.
Yeah, I know. I'm a little bit busy here.
Okay. Remember, though, we're not killing hosts. That would be maladaptive.
Yeah.
Yeah. Okay. Okay, ready?
The fish will be released.
Yes, the fish will be released. Do we need to see evidence of it? I feel like I need.
No.
There we go. Okay.
It's like a blues riff. You got to have resolution at the end.
Yeah. Okay. No. Modified mantle tissue.
Oh, there's still more because this is the thing. Look at this.
This is bonkers.
Look at that. This is part of the body wall.
Yeah.
That if for your body, so this is your skin. This is the outside of your body wall.
Yeah.
Right. This is that skin not just making a shell to protect you from predation. It's a shell that's now going to go, hello, come on over here.
I'm fancy.
So this is not a lure that's advertising. You can't look at this and go, well, there's a species of fish out there and it wants to put its Gluckitia in that species of fish that looks like this, remember. This is an intermediate species of fish that its mantle is mimicking.
Yeah.
The desired Gluckitial host will feed on something that looks like that. And it's doing it. These mussels have no eyes. They have some photoreception. They can tell shadows.
Yeah.
Could you build the model of an intermediate fish species if you could see shadows?
Yeah. No. It's amazing.
How does the mussel know what the fish looks like to replicate it? That's the question.
Okay, super good.
And also into this mix, remember, you always have to stir time.
Yeah.
Not weeks, not months, thousands, tens of thousands, hundreds of thousands of years.
So the answer to that question is natural selection. And when we think about the learning that we've done already about biology, about evolution, we spend a lot of time on natural selection at the expense of all of the other really amazing evolutionary mechanisms. So we're not going to spend so much time on natural selection. So let me just really briefly answer that question, because we're not going to spend so much time on natural selection. So how does the mussel evolve a lure that looks like the food of a fish? And Dr. Smith gave you the answer. Time. It is the slow accumulation of mutations that over time are selected for because they contribute slightly more fitness at each step, right? So the story goes, there's a mutation that increases, you know, the surface area of the mantle tissue by a little bit. And that attracts just a few more fish than it did before. And so that is selected for because the Gluckitia of that individual are able to disproportionately reproduce, right? And that gets passed on. And then within that population or that group, there's another mutation that has little stripies on the end. And that looks a little bit more like the prey of those fish. And so they're the ones that get picked up slightly more than all of the others. And it's over millions of years of this type of story that natural selection does lead to the evolution of adaptation.
So it involves the variation present in the population, that it's heritable, and that there's an overproduction. I'm doing air quotes, Tribbiani air quotes, that there's tens of thousands of young. So if it doesn't work sometimes, that's fine.
Yeah, yeah. So we want you to think a little bit about, for the next time, we're going to do an exercise, maybe not next time, but the time after. Anyway, think about it, because it's not going to be easy. You're going to be like, I can't think of anything. And that's perfectly fine. You can Google stuff. But what we want you to start thinking about is what some of the advantages and disadvantages, what are the pros and the costs associated with evolving such a complex structure. Yep. Because there are some species of eunyonids that don't have that type of complexity. They just have little frilly bits, right? And there's a cost, but there's also a benefit to doing it that way. So think about the costs and the benefits associated with specificity or complexity, and understand that one is not necessarily better than the other.
It depends.
It totally depends. And it's time for your homework, and it's time for us to say goodbye. So
we are in the last minute.
The previous question was your homework. This is also some homework that should be available to you also through course links to be able to get you started. But we covered the eunyonid and the Gluckitia life cycle. Do a very similar thing for the veligers, for the zebra mussels, to understand what is different about them and what the costs and benefits are with respect to their ability to occupy a new space or invade a new area. Why zebra mussels? Why weren't the eunyonids able to kind of hold their ground? And you'll hopefully be able to tie those questions into their life cycle and their natural history.
So be good to yourselves. Be good to your friends and family.
Thank you.
Stay safe and reflect on the fact that you live in a world, like you said in the chat, where mussels bully fish.
Take care, everyone. If you want to stick around for our office hour or student hour, please do. We'll be here in a few minutes.